Welcome, Brookings Honors Bio students. This is Mrs. Rydell, and today we're going to talk about another way that genes are more complicated than Mendel originally thought. And we're going to talk about something called epigenetics. This is a fairly new topic. We haven't research on this didn't really give us information until the early 2000s. In 2003, the Human Genome Project was completed, and that was a major project to look at the sequence of DNA in humans and to try and look at every single chromosome, every little piece of DNA code, and to sequence the human genome. And what we discovered from doing that is that there are way fewer genes than scientists originally thought in the human genome. Somewhere between 25,000–30,000 genes, which is not really enough to code for everything that needs to be coded for to make a human. And so scientists started looking at how is that possible and what's going on with our genome. And what they discovered was some very interesting things about how genes are controlled. And research is learning more and more about the topic of epigenetics. And this is a fairly new field, and one of those fields that's research is happening very quickly now. So let's talk a little bit about how this works. There is a genetic disorder called Prader-Willi Syndrome, and kids who get this mutation are born very weak muscle tone, very sometimes described as kind of floppy, pale. At first they refuse to eat. It's hard to get them to eat. But when they're about a year old, it's almost like flipping a switch in these kids, and they start eating. They cannot stop eating. They never get the signal that tells them they're full. They will eat and eat and eat and eat until they're physically stopped by someone. They will steal food. Kids who have this, you have to put padlocks on all your food cabinets at home, on the refrigerator. They have very tiny hands and feet. Underdeveloped sex organs. They have some physical issues, mildly mentally handicapped, spectacular temper tantrums, especially if they're refused food. They have an exceptional proficiency with doing jigsaw puzzles. That's kind of a weird thing. If any of you saw the movie Rain Man, it was about a man who was autistic, but he had an amazing ability to do math in his head. To figure out calendars and cards and sometimes, even though someone is mentally handicapped in some ways, they have abilities in other ways, and these kids have an ability to do jigsaw puzzles. But the problem with this disorder is, like I said, they can't stop eating. They never get the signal that says I'm full. Most of these people die, middle age from severe obesity, things like heart attacks and things that go along with being overweight. The interesting thing about this genetic disorder is there's another disorder that almost is the opposite physically. There's another disorder called Angelman's. These kids are not born floppy and weak. They're very tight muscle tone, very, very taut. They're thin, hyperactive, small head, severely mentally handicapped. They move in a very jerky, characteristic fashion, almost kind of like a puppet on a string. Very happy disposition. They laugh and smile all the time, even in some situations where you wouldn't think that was appropriate, like when they're in pain. And the interesting thing about kids who have this disorder is it can run in some families with Prader-Willi. And the interesting thing about how it's transmitted is these kids are missing the same piece of DNA, the same piece of chromosome 15 in Angelman syndrome that's missing is also missing in kids that have Prader-Willi syndrome. Some of you may know the actor Colin Farrell. He's in a lot of action movies. He has a son with Angelman syndrome. So they started looking at what's going on and how is it possible that the same missing piece on the same chromosome can cause two very different genetic syndromes. And what they discovered was the kids who have PraderWilli Syndrome are missing that piece of chromosome 15 that came from their fathers on a paternal chromosome. And the kids with Angelman Syndrome are missing that same piece of number 15, but it's missing from a maternal chromosome, came from their mother. So how does a gene remember where it came from? How does it know, a chromosome know that it's a maternal chromosome or a paternal chromosome? And the answer is epigenetics. So we're going to look a little bit closer at this whole new area of genetics. The term prefix epi means above or on top of, and epigenetics deals with what sits on top of the DNA. It's not a change in the actual DNA code. What epigenetics looks at are what are the molecules that sit on top of the DNA that interact with the DNA and control whether genes are turned on and turned off. And so if you look at my DNA here, this white coil, remember DNA coils up very tightly, and when it coils, it wraps around these proteins. These little green circles are proteins called histones, and their job is to wrap up the DNA into chromosomes. And once it's wrapped now, whether the gene is turned on or off, remember we said to unwrap it, we need to open it up to read a code, but if it's tightly wrapped, it's going to be a turned off gene. And what kind of turns off the genes and helps to wrap or unwrap them are these little yellow tags. They are chemical tags, they're chemical molecules that sit and attach to the DNA and control whether a gene is on or off. So genes are actually stamped with an imprint with these tags during gamete formation. So when your body is making sperms and eggs, those chromosomes are tagged as a maternal chromosome, if you're a mom, or a paternal chromosome, and the same genes are tagged in the same way so that all cells in the body are going to express the same version of the gene, either maternal or paternal chromosomes. And the tags that we add, those little yellow markers, are called methyl groups. And so if I look at my piece of DNA here, you can see along the way there are little methyl tags that are added to the DNA that turn genes on if the tags are there, and removing those tags opens the DNA and turns the genes on. And there are many, many mammalian genes that are critical for development, critical for functioning in the body, that are imprinted, that are tagged, and that's what's one of the things that's controlling whether a gene works or not. So the interesting thing, though, about the epigenome is it's not set in stone. Even though the DNA code, the sequence, the A's and T's and G's and C's may not change, what can change are those epigenetic tags, those methyl tags. And it appears that the things that we do can impact those tags, can add tags or remove tags and change our epigenome. So, one that didn't pop. Things that can add or remove tags. Environmental influences. Number one, we know, is diet. Things that you eat can change the tags on your DNA. Exercise can change the tags. Stress level can impact whether DNA is tagged or not tagged, which genes are turned on. And chemicals in the environment, certain kinds of chemicals that affect those methyl groups, can change the tag patterns on our DNA. We're going to watch a little video now. I'm going to ask you to pause this slideshow and go back to the website, and we're going to take a little detour go back to the website, and I'm gonna ask you to watch this little video first before you go on with this slideshow. So go back here, and we're gonna watch this little video. It's called Nova Epigenetics, and I'm gonna have you watch that, and then come back to our podcast. So see you in a minute. Welcome back. Thanks for going to watch the video. What you saw in that video were some of the things that can change your epigenome, and talked about that experiment with the agouti mice, and talked about how those tags change because of environmental influences. With the twin study, how we can see those tags change with age and twins starting to become more and more different. There are many environmental influences that can change those epigenetic tags. So what do we use these tags for? First of all, we use them to tag our maternal and paternal chromosomes. That is how, when we talked about Prader-Willi and Angelman's at the beginning, that's how a chromosome knows it came from mom or it came from dad, because of those chromosome tags. It's important in embryo development, differentiation. How does a cell get to become different? Remember we talked about the way you make a brain cell or a muscle cell or a bone cell or a skin cell different from each other, is that we're turning on and off different genes. We don't want lung cells making toenails. We don't want skin cells making eye color genes. And so in those cells, as those genes get turned off with the epigenetic tags, they're permanently shut down, and that's what makes the cell differentiate and become a different kind of cell. Anytime you have changes in how cells act, you're going to have epigenetic tags involved. So when things click on or off during puberty, it has to do with changing the tags, which genes are expressed or not expressed. We are not changing the DNA sequence, we're changing which genes are turned on or off. Pregnancy, different genes are turned on that aren't on during other times. During aging, and we think this may have something to do with aging of cells, which genes get turned on or turned off. Differences in the tags can also lead to some diseases. If we're turning off genes that are important for the cell to function, we can have some diseases like cancer, or lead to genetic disorders like we talked about Prader-Willi or Angelman Syndrome. Those can be problems caused by changes in epigenetic tags. So let's look a little bit more closely at how this works. When a cell, a gamete-making cell in an ovary or testes, is going to make gametes, make sperms or eggs, right? If you are a mom, you have two chromosomes. One came from your mom and one came from your dad. They're tagged as maternal or paternal chromosomes. And what happens now is those are the ones you inherit, but if you are a mom making eggs now, you need to tag all of your chromosomes that are going on to your children as maternal chromosomes. Or if you're a dad, you're going to tag them as paternal chromosomes in the gametes, because that paternal chromosome now is going to go on to make a new person. So when gametes are formed, the old tags are erased, and all chromosomes are re-imprinted according to the sex of the individual that is making the sperms or eggs. And that imprinting is critical. It's important for normal development. And what we know now is that many of the tags of which genes are on or off, when they're re-imprinted, they're put back on in the same way that you received them. And so things that tags that you inherited from parents or grandparents even are still showing up in the chromosome imprints on your DNA. Epigenetic tags are important in development, turning on and off different genes at specific times. There's an important link to cancer. One of the genes, we call it the breast cancer gene, is basically tumor suppressor gene. It's called the BRCA gene, and we have two of them, BRCA1 and BRCA2. This is a tumor suppressor gene, and people who have a mutation in this tumor suppressor gene, basically it's a mutation in the whether the gene is turned on or turned off. And its job is to suppress tumors, to block the tumors and stop them. So if this gene is turned off, it's going to become a cancer cell. We don't repair the damage, we don't block the tumor, and you can have tumors starting to grow. So whether it's off or on has to do with those epigenetic tags. Other important things to think about in this area, this topic raises so many questions. There's tons and tons of research. There's some links off the webpage if you're interested in learning more about this. But things to think about. So what? What does this have to do with you? The first thing to note is that this is not a mom thing. This is not an environmental, something happens to the baby growing inside the mom because of things a mom is eating or the mom. That is part of it, but it happens also in dads. The things, the choices that you make, the things that you do impact your epigenome. It changes your methyl tags. And so even guys, what guys do, you wouldn't think that that would impact their babies, but it does. There are studies that say that guys that start smoking as teenagers are more likely to have sons that are overweight down the road. There's are studies that show that what your grandparents ate and whether they lived in times of famine or times where there's food available impact health issues in their grandkids, not just their kids, but their grandkids. These are tags that are passed through families over generations. Is it possible now that we know how these tags work, to come up with some therapies to treat some of these disorders? Can we purposefully change our tags? And there was a little section in the video that talked about doing epigenetic therapy for cancer patients. Think about this. If the things that you do can not only impact you, but impact those tags that are going to get passed to your kids, even to your grandkids, how does what you do impact your epigenome? And what responsibility do you have to protect your epigenome and think about the impact that you can have on future generations? This is a whole new area, and we're just beginning to understand how this works and the implications of this. But it's something to be thinking about for the future. Thanks for watching.